Welcome to the Garvan Institute of Medical Research for our first public seminar of 2026. I would like to begin by humbly acknowledging the traditional custodians of the land on which the Garvan Institute is located, the Gadigal and the Bidjigal people of the Eora Nation. I'd like to pay my respects to elders past and present and acknowledge their ongoing connection to land, water and community. And also I'd like to extend my respects to any First Nations guests who may be joining us today. Now, we are all here because we share a common interest and passion for science and its ability to advance human health. I think that's not up for debate, and I think the sheer number of people that are in this auditorium here alone is a testament to our engagement for today's topic, which is arthritis. And honestly, I think you would be hard-pressed to find anyone who either is not themselves affected or knows someone who is affected by this condition. In Australia, it affects 1 in 7. So here at Garvan, we have the Masle-Farquhar lab and our biologics platform, who are working hand in hand to develop diagnostic tests and new targeted treatments for this condition. And today we have the privilege of hearing from some incredible speakers, including Dr. Masle-Farquhar himself, Dr. Mukta Bhati, who is a lead scientist within the Biologics Platform, and Mr. Kevin Nafty, who is an advocate and is using his story to promote awareness of this condition. Thank you so much, everyone, for being here today.
Hello, everyone. This is very exciting. I want to thank you all for being here. I'm very privileged and very excited, actually, to stand here and talk to you today, because it's an opportunity for us to tell you about really the culmination of 4.5 years of work in launching and rapidly growing an arthritis research and clinical translation program here in New South Wales and across Australia. And I really hope that this can stimulate more conversation and more energy around doing new things in arthritis. All right. 1 in 7 Australians have arthritis. Now, Viv mentioned this just a second ago. What does that mean? Well, there are many forms of arthritis, but they all cause chronic pain, irreversible joint damage and lifelong uncertainty. And you can see some of these manifestations listed up here. They increase risk of lung, heart, cardiovascular and other diseases. And they really, and I want to emphasize this, they cause major psychological and financial distress on not just patients, but also their families. And I think that's something that's really under-recognized in our community in the way that we think about arthritis and its impacts. And it's a staggering cost to Australia. Every year, $6 billion spent on arthritis. Now, to give you some scope of what that means, that's the equivalent of paying salaries for 65,000 new nurses in Australia every year. Now, arthritic diseases currently have no cure, and really what that means is that these are chronic and lifelong conditions, and we really can't predict who will respond to treatment, how, or when. Now, I want to give you an example specifically what that can look like. It's easy when you hear large numbers to get lost in the statistics, but what does that mean for people in their day-to-day? This is the journey of one woman with very treatment-refractory psoriatic arthritis. She came into the clinic with severe right ankle pain at the age of 25, and she had to undergo a biopsy, a whole series of blood tests and imaging, and was eventually diagnosed with seronegative peripheral spondyloarthritis. Now, this here lag between July and September in getting a diagnosis is actually quite lucky. Many people with arthritis will need to wait months or even years for a diagnosis. Now, at this point, she was put onto what we call disease-modifying anti-rheumatic drugs, or DMARDs. These are first-line therapies, and so in this case, methotrexate and sulfasalazine. Now, in March 2019, it became apparent that this was not working, and her arthritis had spread at this point to more than 5 joints. And so they put her on a much higher dose of prednisone, subcutaneous methotrexate, secukinumab, and leflunomide, which are another series of drugs. And again, by January 2020, we can see here that her arthritis has spread now to over 20 joints, and she was having bloody diarrhea and a number of other complications. At this point, she went onto another dose and round of subcut MTX, or methotrexate, tofacitinib, leflunomide, etanercept, and the picture at the end of 2022, at the point this paper was published, was that she was still not responding and her hips were heavily involved. Now imagine this is her at 25 and this has been her life for 5 years. Now, I want to point out to everyone that not everyone's journey with arthritis is like this, and there will be people who are affected at different ages. There will be people who are lucky and who respond well to these first-line therapies like methotrexate and sulfasalazine. But I want to emphasize that those good responders are by no means, wholly lucky and wholly cured, in the sense that these people still have to deal with a lifelong diagnosis, lifelong treatments, and management of a chronic condition. And I think it's too easy for us to shrug off in the people who do respond that problem is solved. And Kevin and other people in the audience can tell you that this kind of story of having to cycle through a whole round of different biologics and treatments is by no means unique. Now, what does it mean that 4 million Australians have arthritis? This is what it means. It means essentially the equivalent of the populations of WA, Northern Territory, and a large part of South Australia are all dealing with arthritis. And this is not including their families and their patients and their loved ones. By the time I finish this talk, actually, 5 Australians will be diagnosed with arthritis. And by the time we finish the Q&A after that, another 5. And really, this is an incredibly urgent, unmet need that we need to be thinking about more, talking about more, and addressing more. And so really, this is what our lab and our series of clinical and scientific collaborators are focusing on. It's really, how do we tackle the clinical unmet needs? And we start with the unmet needs, and we derive our mechanistic and scientific questions from those. And I think that's the best way to derive impact and to make a difference in our community with people who have arthritis. And so the goals appear quite simple. They're very hard to do, of course, but the first is to reveal disease mechanisms, accelerate diagnoses, predict and suppress flares, and we'll be talking a fair bit about that today, but also identifying biomarkers to guide treatment choices. I think many of you here in presence or online will be aware that a major problem right now is that we're treating many people as what we call best guess, in the sense that we don't currently have good molecular or cellular guides to help clinicians select the best drug for a given patient. We have no means of predicting which drug you will or will not respond to. And finally, we're developing novel and more effective, hopefully, therapies. So how do we go about doing this? Well, one of the key answers, at least from my perspective, is what we call immunogenomics. Now, immunogenomics is the study of immune cells and disease mechanisms in great detail using genomic technologies. Now, these are cutting-edge, really quite revolutionary methods that have changed the way that we can do medical science, particularly over the last 10 and much more so over the last 5 years. We can really do things now that were just not possible previously. We can gain insights in a way that was not possible previously. And so the outline of today's talk really follows the following set of stories. The first is quite a brief outline of the work that we're doing in osteoarthritis. And in that context, we're using immunogenomics to do a really unprecedented dive into the cells that are present in the joints of people with osteoarthritis. I'll then talk in more detail about what we're doing in the context of rheumatoid arthritis, and specifically of rheumatoid arthritis flares, and the role of these type of immune cells that you're going to hear a lot about, which are called killer CD8 T cells, and how this work is actually directly informing our collaboration with the biologics facility, including Dr. Mugdabati, and our design and very imminently now preclinical testing of novel biologics. And finally, I'm going to give you a brief overview of what I view as the current Garvan Arthritis program and where I think we need to take this over the next 5 and 10 years, not just at Garvan, but collectively, and how we've gone from, one person with a couple of ideas 4.5 years ago to over 40 collaborators, over 15 hospitals and institutions around Australia, and now collaborations with France and Finland and the US, and from 0 to 400 participants around Australia. So in the context of osteoarthritis, what are we focusing on? In this context, we're focusing on the very variable levels and unpredictable levels of pain, the lack of available biologics, and our strong reliance right now on joint replacement surgeries. So more than 500 million people worldwide have osteoarthritis, and it's associated with a whole host of metabolic, cardiovascular diseases, osteoporosis, reduced activity, accelerated cognitive decline, reduced quality of life, and a big impact on mental health. Now, what's really important here is that OA causes very highly variable stiffness and pain levels, and we currently don't actually know how to predict those. I think this next statistic, to me, is astonishing. There are more than 500 million people affected, and we currently have no OA-specific disease-modifying drugs approved clinically. I think that's just incredible. 1 in 3 people with knee OA will require that knee to be replaced. So one of the things that we've done is we've set up a really close and deeply integrated collaboration between the lab and these 2 major partners, as well as a whole host of other people. Nick Versailles and Sean Mungovan, who head the orthopedics and allied health service departments at the St. Vincent's Clinic next door. Every year at the clinic next door, there are 1,000 hip or knee replacements for people with arthritis. And this is actually an underutilized resource that we can use to better understand and treat arthritis. And I want to really point out several of the key members in the team who've been doing a lot of the hard work in terms of biobanking, collecting clinical data, and investigating these samples. But what does this look like? From these joint replacements, these knee replacements and hip replacements, we're now collecting synovial fluid, which is a liquid that accumulates around your joints and lubricates them, but accumulates when you have inflammatory flares. We're collecting synovial tissue that's often degraded in arthritic diseases, bone marrow where your immune cells are actually developing in the first place, bone which can be affected by osteoporosis and various metabolic diseases, blood, saliva, detailed imaging data, and from every patient also comprehensive physiotherapy assessments of pain, stiffness, quality of life, not just before surgery but also after surgery. And I want to highlight here that all of these tissues are normally discarded. And the reason for that is that they're not part of a research workflow in the hospital, and that's quite natural. But this is an incredible resource for us to understand arthritis more effectively. And so what we've done here is repurpose and rescue these samples that are normally discarded for research. Now, I apologize for anyone who's squeamish, and we'll go past this slide pretty quickly. But I wanted to give you an idea of what this looks like. On the left here, you have a tibial plateau, which is taken from a knee replacement surgery. In the middle is the synovium, which is this tissue that's often attacked in inflammatory forms of arthritis. And on the right, we have half of a femur head that's actually taken during a hip replacement surgery. Now, from many of these individuals, we can gather very quantitative data on their range of motion, their stiffness, physiotherapy outcome measures, but we can also get self-reported outcomes on the degree of pain, stiffness, and impacts on quality of life that these people are experiencing. What does that look like? This is a summary of these kind of data for over 100 people with osteoarthritis, and we're gathering these for essentially all of the participants going forwards. What this allows us to do is to take a really clear picture at the severity of osteoarthritis that's being experienced by all of these participants. And more than that, we can now start to select, because we have the size and an amazing biobank that's continually growing, we can select the extremes, and we can start to ask, what is happening in the joints of these people who have the most severe levels of pain relative to people who have no pain? Are there differences in the immune cells or other cells and molecules within their joints? What we can also start to ask is, does osteoarthritis differ between different joints? In this case, hip versus knee, and you can see the two different colors here. Essentially here, every dot is an individual patient, and we've collected data from a hip or a knee replacement surgery, and we can quantify and look at in detail at the phenotype and features of immune and non-immune cells in the joints of people with these different joints being affected by osteoarthritis. Now, as of recently, we've actually also started collecting ankle surgeries, and the next stop is shoulder surgeries. This is going to be an incredibly precious resource, not just for Garvan, but really, I hope, for scientists and clinicians across Australia. And so I don't have time today to tell you more about some of the insights that we're trying to make in osteoarthritis, but I want to highlight these concepts because they're true for osteoarthritis, but they're also true for what we're doing in rheumatoid arthritis, gout, psoriatic arthritis, and so on. Clinic lab needs to be integrated. We need the scientists and the clinicians to be talking, to have direct access to one another, and to be directly informing each other's work. And in this context, we have direct and unfettered access between the lab, the surgery theatre, and the rehabilitation and allied services. What we're doing is recycling tissues that would otherwise be discarded, and we think this is now the fastest growing osteoarthritis biobank in Australia. And we're also applying novel technologies that we specialise here at Garvan. What they allow us to do is to study immune cells and genomics in incredible detail, in this case, as an example, from over 300,000 cells isolated from the joints and blood of people with osteoarthritis. And so on this front, I ask you all to stay tuned, but maybe the next time we get together, we can talk about osteoarthritis. Okay, so the focus now shifts to what we're doing in the context of rheumatoid arthritis. And in the context of rheumatoid arthritis debilitating flares, the roles of these killer immune CD8 T cells, and how we're going about trying to target these cells now with new biologics. So what actually led us to study rheumatoid arthritis in the first place? Well, the reason is actually quite simple or quite elegant to my mind, and that is a striking genetic association between rheumatoid arthritis and this gene that you can see here called STAT3. Now, I don't expect you to remember this acronym, TLGL, but it's a rare cancer of your CD8 T cells. What's striking about TLGL is that in 1 in 2 cases, those TLGL will acquire a mutation or a DNA change in this gene called STAT3. Now, if we look in the general population, 0.5 to 1% of people will develop rheumatoid arthritis, roughly 1 in 100 people. Now, if you have this TLGL rare CD8 T cell cancer with no STAT3 mutation, it jumps to 6%, or more than 6 times the increased incidence. But if you have more than one STAT3 mutation in that cancer, suddenly 43% of those patients will get rheumatoid arthritis. So more than 40 times the expected incidence of rheumatoid arthritis in the general population. And what that does is it gives us a clue that there is a striking genetic association here that points to a possible role of CD8 T cells and of STAT3 mutations in the rheumatoid arthritis that affects millions of people worldwide. But there's a critical question, and that is, do these STAT3 mutations and these CD8 T cells actually cause inflammation and disease? Or are we finding them in inflammation and disease because they result from inflammation and disease? And that's a critical distinction. Now, we won't go through it today, but this is actually directly what we showed in our 2022 immunity publication, that yes, indeed, STAT3 mutant CD8 T cells can cause disease. And we did that by studying a variety of different methods, but really by leveraging detailed study of rare patients, in this case, unfortunate children with inherited mutations in this gene STAT3, really carefully designed preclinical models and some of the cutting-edge technologies that I'm telling you a little bit about today. And collectively, what that allowed us to do is to show the causal role of these cells in that disease, but also to identify the mechanisms by which we think that is happening. Now, I'm a massive nerd, in case you hadn't realized. I'm actually only putting this figure up because I love the figure and I spent a long time working on it, but you might see it again throughout the talk. Now, the question is, what about rheumatoid arthritis now that we've showed this causal role, but what is happening in patients with rheumatoid arthritis? The hypothesis here is this: driving rheumatoid arthritis flares. Now, flares are a massive unmet clinical need. Flares are essentially severe, debilitating, and unpredictable worsening in the condition of arthritis. Now, what that can look like is this picture on the right here with someone who has a knee effusion with an incredibly swollen joint that requires draining to alleviate their pain and inflammation. And that is something that would happen quite routinely in clinics around New South Wales and Australia. Now, currently, and I pointed to this earlier, treatments are selected by best guess, and this is not a problem of clinicians. There are incredibly amazing clinicians, including in this room. The problem is that we don't have cellular or molecular guides right now to direct treatment. We know now that less than 20% of patients currently are entering what we call sustained remission, and this to me is very striking. 2 in 3 patients with different forms of arthritis will experience flares despite being on treatment, in some cases despite being on more than a decade of treatment. And what often also happens is that someone's symptoms and their severity will start to taper, or let's call it ameliorate. And what will happen at that point is that the clinician will want to reduce the dose of drug being given to those individuals because they have side effects. What often happens in that context is that that is when flares strike, and that the severity of the symptoms worsen, and suddenly the disease is actually no longer controllable with the same dose of drug being used previously. So instead of decreasing the dose of drug being given, you're having to increase it. Finally, each rheumatoid arthritis, psoriatic arthritis, and gout flare is associated with increased risk of cardiovascular or heart disease. And just to point out, one relevant statistic here that is, incredibly important to my mind is that people with psoriatic arthritis are up to 1.5 times more likely to die of heart disease, and we don't fully know why that is. So what we've set up over the last few years is a network primarily around Sydney, but looking to expand slowly. The logistics of this are very complicated, such that within 2 to 3 hours of your hospital visit, going as far afield as Westmead or down south in Wollongong, we will get a call if you are getting your joint aspirated, and we will get that synovial fluid and paired blood into the lab, biobanked it, stored, and experimenting on it. And that's what this growing biobank of joint aspirates at time of flare looks like. Now, I just want to outline for you down here on the x-axis these different forms of arthritis that are represented by different colors. And the point of this is to show you that we are collecting these samples from people with different kinds of arthritis, and we are doing this very much on purpose, because by comparing different forms of arthritis, we can start to pinpoint what is different about rheumatoid arthritis versus gout versus psoriatic arthritis. What are the disease-specific mechanisms? This is really important. What we're now really starting to get, and this is something I'm incredibly excited by, and I think it's critical, is sequential flares. Now, what that means is that this is individuals with poorly controlled arthritis who are experiencing multiple episodes where their joints require draining, and that we have set in place sufficient logistical and staffing and infrastructural considerations around them that we are managing to collect samples
from their aspirated joints, not just once, but twice, 3 times, or even 4 times. I'll tell you a little bit more a bit later about why that is so important, but I actually think that sequential flares are going to be really critical to understanding RA flares, and the same is true for other arthritic diseases. Now, how do we identify rare or abnormal cells that are driving flares? The answer is similar to what I was pointing to earlier. It's the application of immunogenomics methods. Now, we can apply these methods to these synovial fluid aspirates from people at time of flare. And this is a schematic of what that can look like. Essentially, we can study at unprecedented detail thousands of cells that were taken from the joint at time of flare from each individual, their paired blood, and from each of these cells, which in this case is represented by all of these colorful dots here, we're measuring thousands of genes within each individual cell. This is something that was simply not technically possible 10 years ago, and to some extent even 5 years ago. What that looks like in reality here is quite amazing data sets. So this plot here shows you about 100,000 cells that we've removed and studied at incredible detail from 30 aspirate samples taken from people with different forms of inflammatory arthritis. And by measuring thousands of genes per cell, you can see here that we get incredible resolution to identify the many kinds of different cells that are highly heterogeneous, that have different functions, different features that are within your joints at time of flare. And what we can do is we can apply unbiased analyses to try and figure out what is going wrong, what is different in these samples. And what is very interesting and exciting to me personally, I suppose, is that killer CD8 T cells actually represent a really significant fraction of the cells that are accumulating within inflamed arthritic joints at time of flare. These are the same cells that really resemble the ones that we think are driving flares. Now, we can zoom out on this picture and zoom in very specifically on these cells of interest, given the huge number of features and measurements that we're able to make now with these new technologies. And one of the things that we're able to do is to assess what we call expanded clonotypes. And I want to profile, not just with this result, but with many of our analyses, a pretty amazing scientist called Catherine Jackson, who's leading a lot of this work. And really, what these methods allow us to do is to identify what we call expanded clones that you can see here in blue. Now, the full detail of what these cells do is sort of too complicated to talk through in this short talk, but notice how all of these blue expanded clones are really clustering in the bottom here, which corresponds to the bottom quadrant of where all of these killer CD8 T cells are. And so essentially what we're finding is the accumulation of killer CD8 clones. And what we mean by clones is that all of these cells share the same cell surface receptor, meaning that they are actually recognizing the same molecule. Now, we don't know what that molecule is yet, but a strong possibility is that it's a molecule that's expressed by the cells in your joint, and that is leading these CD8 T cells to attack those joint cells. So I've told you now that killer CD8 T cells are present and quite abundant in inflammatory flares in the context of rheumatoid arthritis. We can also find that they're actually expanding as clones in joint fluid during these flares. And the next question is therefore, well, what is actually happening in sequential flares? Do these killer CD8 T cells persist during sequential flares? Now, why do we care? Why is it important whether cells persist during flares or not? Well, the reason for that is that when you're studying an inflammatory episode, whether it be a joint aspiration, a bronchoalveolar lavage, what have you, you're looking at someone who's undergoing a really systemic inflammatory process in a lot of contexts. And so when you look at a single point in time, how do you distinguish which cells are dysregulated or abnormally there because you have systemic inflammation versus the cells that are causing that inflammation and process in the first place? By contrast, if you start to find the same population that's not supposed to be there and that's there every time you have a flare, you can start to become a bit more confident that maybe these are the cells that you're looking for. And that's what this looks like. A small number of individuals now where we're capturing these sequential flares and we're actively trying to capture more. What this graph actually shows us on the left is that specifically in rheumatoid arthritis, but we don't see that in gout, and this is consistent with these diseases having very different mechanisms, we are finding that these killer CD8 T cell clones do persist between sequential RA flares. You can see it quite nicely if we focus on this participant RA1 here, that in these 3 sequential flare aspirates that we've collected from this person who had poorly controlled rheumatoid arthritis, every time we collected them, we find these killer CD8 T cell clones in their joints. Now the question is, can we detect persistent killer CD8 T cells from the joints in the blood? And if we can, do their levels actually vary between flares and remission? Now, why is this important? It has immediate implications for disease monitoring, For flare prediction, potentially, if you were to find that killer CD8 T cell clones are increasing in the blood prior to someone experiencing flare, could you get an advance warning that this is about to happen? And really importantly, for rapid and detailed assessments of how well someone is responding to a therapy, such that if you have rheumatoid arthritis and if we can demonstrate the causal role of these cells, and there's a lot of work that needs to be done, but that we might get to a point where rather than getting a new therapy when your previous therapy wasn't working and having to wait 3 months, 6 months, or 9 months to find out whether your body is telling you that your drug is not working for you, you could actually take routine blood tests and monitor these cells in live time. And it would allow clinicians on a rational basis to rapidly switch you to a more effective therapy if they thought that was required. Now, I've given you probably a clue in asking all these questions because I'm giving myself easy answers. It's usually a yes. But in this case, what we were able to show is that yes, indeed, these killer CD8 T cell clones in the joints are detectable in the blood. Now, these graphs are a little bit complicated, but what they basically show us, and I wanted to highlight that this is true for different cell types in rheumatoid arthritis, but also psoriatic arthritis, which I think really points to the utility of what we're doing in not just one form of arthritis. What they show us is that the most expanded killer clones, the most highly proliferating ones that we find in the joints, which are in these blue crosses here, are also the most abundant clones that we find in the blood of these same patients. And this result is really hot off the press. This is as of 2 weeks ago, and we're really excited by this. So we've shown you that these killer CD8 T cells are persisting every time these people in this small cohort of people with RA have flares. And we're showing you now that we can actually detect them in blood at time of flare. Now, the next question is, do they decrease in blood at time of remission or of non-flare? That's really critical. That's underway, and we expect to have answers in 3 to 4 months. That will have immediate implications for how we might use these cells to monitor disease and better understand disease. And finally, and this is where Dr. Mugdabadi and some of their team come in, how do we, in an incredibly precise and targeted way, attack these killer CD8 T cell clones with new biologics? And so this is where Rachel Galamidi and her team are really leading the foray. And this was an incredibly exciting part of being at Garvan for me, was when this biologic platform was launched. And it's taken sort of 2 years, and Rachel came over from the Scripps Institute in the US, where she led all the way from design to commercialization, a whole series of different molecules. So we have the expertise of Rachel and people like Mugda who've got, incredible expertise in biologic engineering, commercialization, and all sorts of key aspects of how you might design and test new therapies here at Garvan, helping to really actively target the cells that we think might be causing disease. And so over the last roughly year, we've been working in really close collaboration, in mutual discussions, and in co-design. And I'm really excited because in March, as of essentially the last few days, this is finished up. The first set of candidate bispecific antibodies has actually been produced to hopefully target these killer CD8 T cells. And so the incredibly exciting timing here is that as of this month and next month, we will start preclinically testing these novel biologics, which have derived from this program of work. So I haven't quite finished yet, but I wanted to take a brief pause before I do finish to thank many of the people who are involved in this work. And you can see now that we've gone from, one person with quite harebrained ideas to a whole series of people who are working very hard and passionately to make this work, and so much so that there's not enough room on this slide now here. But I really want to thank also the funding bodies that have made a lot of this work possible, the philanthropists who actually, really without which this program never would have come online. It just simply wouldn't be possible. A big thanks to Kevin for all of his help and support, and looking forward to our discussion in a second. And in particular, I want to start with my team, and many of them are actually in the front 2 rows there. Incredible people to work with, so if you get a chance to shake their hands. And then really to the patients and their families who are donating generously their samples, their time, their enthusiasm to help us with this research and trying to change the way we think about arthritis. And so I'm going to finish up with this next couple of slides, and I really would want to just reiterate this point that 4 million Australians have arthritis, that in the time I've just been talking to you, another 5 people have been diagnosed and their lives have changed. Another 5 will be diagnosed when we have our discussion in a second. And what we really need to be doing now is leveraging this recent revolution in immunogenomics to start to change the way that we study and thereafter treat arthritis. We can start to really identify disease mechanisms, generate new treatments, and try to target existing treatments far better. And that's an angle that we can also talk about in the Q&A. And so really my vision for this is that we need to establish here an international hub for arthritis research and clinical care. And part of what that will mean is, in fact, that we will train the next generation of clinicians and rheumatologists in immunogenomics, so that going forwards we have more and more integration of these processes between clinics and the lab to use new technologies in the day-to-day to try and improve the way that you're all treated and that we're all treated. That we will develop more effective therapies, that we will actually identify biomarkers, and this is what I just touched on, that actually allow finally informed and rational selection of the most optimal treatment for every individual based on their individual genetics or other biomarkers. We need to increase community awareness of arthritis and change the way that we talk about arthritis and talk about arthritis more and chronic pain and the impacts that it has. And we need to model and fund and start to establish a long-term centre here for multidisciplinary care and research that are integrated. And what I see is happening is that over the next 5 to 10 years, if you have arthritis and you have heart disease, or you have psoriatic arthritis and you have skin disease, that when you go into the clinic, you would be seen by multiple specialists working together, not sequentially, in an integrated way, and working with the researchers directly, such that in real time, we can start to move faster with the way that we treat arthritis. Now, that probably sounds wild and crazy, and in fact, it does seem wild and crazy. I suppose one thing that's worth telling you all is that during this whole journey, about a year after I started this arthritis program, I myself got arthritis. And, you know, I won't go into all of the details, but I couldn't stand up in the shower for a year for the pain. I had chronic pain for 3 years. We need to be doing more now, and you know, I think that gives me some sense of what some people are going through. But fundamentally, I've been incredibly lucky, and there's a huge number of people out there who really need us to do better and faster. And so this is what we've managed to do in the last 4.5 years, is establish this hub that we're starting to grow here at Garvan for arthritis research. And I've told you about osteoarthritis and rheumatoid arthritis. I haven't touched on what we're doing in psoriatic arthritis and gout, and some of what we're trying to launch now in consumer advocacy, in clinical engagement, clinical education. And really, I want to end here on this simple note, actually, which is that I'm really happy to be here, and I'm a nerd, so I could blab to you for hours about what I do, and you're going to get incredibly bored. But fundamentally, why I'm here is also to tell you that you can help. The people who are online or in live here, we can all contribute in different ways. You can support Garvan, you can support another organisation who does arthritis work or clinical research. You can talk to patient advocacy bodies like Arthritis Australia, Movement Australia. You can talk to your MPs about getting more funding for arthritis care. If you're a scientist or a young medical trainee and you care about arthritis research, you can come to the lab and work with us. And I think fundamentally what we also all need to do is check in with our family and friends who have arthritis and do that more often. So with that, I just really hope that our discussion now, we can use this as a time to discuss how we can make things better and faster for arthritis. And thank you for your time. Looking forward to talking to you all.
Just as by way of introduction, Mugdha is a lead scientist in the biologics team here at Garvan. Now, give us a little bit of a 101. So Etienne just mentioned biologics. What is a biologic? What does it actually do? And why is it so exciting? We hear the term quite a lot these days.
No, that's a great question. So unlike your traditional drugs that we all know very well, like your Panadol, etc., other small molecule drugs that we're all familiar with, small molecule drugs are broadly acting in the body. They don't just target one specific tissue type, for example, and for that reason, they can have various side effects. They can be toxic as well. Biologics, on the other hand, I like to think of as a more like a precision tool. They act very precisely on specific parts of your body. And for that reason, we can actually design them to target specific cells of the immune system, such as those that Etienne has described, for example, or particular molecules. We can deliver them to very specific parts of the body so that they're actually just acting on the particular problem that we want to abrogate. For that reason, biologics give us better disease control and hopefully with fewer side effects to patients.
Incredible. So basically biologics are super drugs in a way.
That's it.
Why are they so well suited to arthritis, to the problem that Etienne is looking to address?
Yeah, so because we can target biologics to act on very specific surface molecules of immune cells, such as those that Etienne has described, we can target particular cell types, such as the populations that Etienne has described that have a role in different types of arthritis or are upregulated in different types of arthritis. We also know that in arthritis, the immune system is not just overactive, but it can be misdirected. And we can actually target both types of pathways in arthritis and have biologics intercept or interrupt specific components or steps within those pathways to reduce their activity. And therefore, in doing that, we can actually reduce the harmful inflammation and, again, offer better disease control and quality of life for patients.
Brilliant, thank you. And just finally, can you give us a little bit of an insight into how you work with each other? How the discoveries that Etienne's team is making in the labs translate to the work that you're doing in the biologics platform?
So firstly, Etienne's team has done a phenomenal job in identifying various biomarkers of different types of arthritis. In those types of discussions, we have collaboratively identified how we could perform translational discovery. So again, Etienne's team, has given us a lot of information on the background of the diseases, the molecules that work together to cause flares, etc. And what we're doing now is, having taken that information, we have designed molecules that can target multiple different types of, the harmful inflammatory markers or inflammatory signals in the body. And we're working on creating molecules that can target multiple components of the immune system to control their activity better and reduce flares.
Thank you so much. Would you like to add to that, Etienne?
I was thinking potentially there was a risk that Mugdha was going to say that we annoy you a lot and hassle you a lot. But we work together incredibly well, and I want to share something that I think is really exciting about working with the biologics team, which is that, we specialize in the lab and many of the scientists at Garvan in trying to uncover disease mechanisms and novel targets and making insights into disease. But what Mugdha and the others do is bridge the gap between those discoveries and our ability to do something about it. And that's not something that we specialize in or have much experience in. And these guys are the best at what they do. Some of the discussions that we have with, Mugdha and Rachel are very nerdy. But a lot of ideas get thrown around that, the sky is kind of the limit in terms of what could be tested in the long term in arthritis. It even goes to improving drugs that are currently out there. If you're given Humira and it's not very effective, can we modify Humira with another component that directly targets Humira to your affected joints? Things like that are not out of the realm of possibility. And so I spend a lot of time just listening to the team and kind of admiring the things that they can now make possible. It's very cool.
Thanks, Etienne. That was much nicer than I thought it was going to go. But yeah, as Etienne mentioned, that's why the biologics team is here. With the up-and-coming biologics and what we know, with further research and understanding of arthritis, we can design the next generation therapies to target very specific components of arthritis and different types of arthritis for better patient outcomes.
Thank you. Kevin, I'd love to go to you now. Thank you so much for being here. You are an advocate. You were diagnosed with arthritis in your 20s, I believe, and you have actually been involved in this research yourself. I'd love to get to that, but before that, I'd love to for you to take us on your journey. What was the process of your diagnosis, and how has your treatment trajectory gone from there?
Sure. Well, first up, thank you so much for having me, and it's been an amazing experience participating in this research, and I'm looking forward to seeing where this goes. I was diagnosed with psoriasis in the first instance, probably when I was about 17. And then in my early 20s, I discovered actually in my toes, I started feeling really stiff, a lot of pain, and started getting that kind of fat toe. That's when I was diagnosed with psoriatic arthritis. At that time, I was actually living in Israel. And just as the months went by, more and more joints started getting the same symptoms as that initial toe. They put me on different, I think I started with methotrexate in the first instance. And then, luckily enough, with my dermatologist, my skin doctor in Israel, she had told me about a clinical trial that was starting up for a new biologic called secukinumab. And I jumped into that to actually participate in that. And at the time, it was just for psoriasis. It had this trial, was focusing on that. It wasn't even focused on the arthritis. But within the first few months, my symptoms, my skin, my psoriasis, and my arthritis symptoms had cleared up amazingly. I stayed on that trial for 2 years. And then probably only a few years later, it got approved for arthritis and psoriatic arthritis as well. I ended up staying on that drug for probably over 10 years. It got a bit complicated times when I moved countries. I moved to the US from Israel and then moved to South America. In different countries, obviously that drug might not have been registered. In the US, we know how complicated that health system is. The insurance wouldn't cover me. They told me that, I skipped all the other alternatives before jumping to the latest biologic. But finally ended up getting it for compassionate use directly from the pharmaceutical company. And then probably around 2 years ago, I actually relocated back to Australia, and just before that, the secukinumab just stopped working. So it was amazing for me for over 10 years, and then just stopped working. Luckily in Australia, being an Australian citizen, I had access to the PBS system and had access to really good rheumatologists and dermatologists. And literally over the last year and a half, I've cycled through 8 different biologics and targeted therapies, each one having to try for 2 to 3 months before seeing whether it has an effect or not, and at the same time being on prednisone, cortisone, and so what would happen is we would get to a point around 2 months being on the drug, we'd start lowering the prednisone to see actually if the biologic was helping, and I would have these huge flares because the medications weren't holding, would have to increase the prednisone again, and then try the next drug. But I'm happy to say actually that as of 1 month, the last biologic that I've just tried is finally working. So it seems like we found the one. I'm almost off my prednisone, and really hoping that this drug holds for the next 15, 20 years.
Amazing. Thank you so much, Kevin, for sharing your story. That's incredible. I understand that you have been involved in Etienne's research. You've donated some synovial fluid to the biobank that Etienne mentioned. Can you give us a little bit of an overview of what your hope is for this research?
Yeah, so I guess firstly, I'm always been open to participating in this research. I participated in a clinical trial quite a few years ago. I've experimented with new and alternative medicine, been involved with medical cannabis also for the last 10 years, hoping to find some solutions there. With this research, I'm excited just at obviously working alongside Etienne and his ambition and his expertise and the surrounding teams as well. I think for me personally, solving the issue, the problem that I've had over the last year or 18 months, whereby I've had to cycle through 8 different biologics, going about trial and error, now having experienced that problem, I wouldn't wish that on anybody else. So really being a part of this research and, solving that issue for many other people is critical. And just, as Etienne mentioned, with all this new technology available, I'm excited to see what else can come out of this, what other target therapies. There's probably a whole host of solutions that we haven't even thought of that's going to come out just with the accelerated technology and, AI, immunogenetics that they spoke about. So super exciting times to be involved in research.
Finally, just a really big thank you to everyone who joined us today. We couldn't do any of the work that we do at Garvan without the incredible support of our Garvan family. Thank you so much for your continued engagement, and we hope to see you back in the building and online very soon.